Welcome to Flow Stars, candid conversations between Dr. Peter O'Toole and the big hitters of flow cytometry, brought to you by Beckman Coulter at BitesizeBio.
Hello, I'm Peter O'Toole, and today on Flow Stars, I'm joined by Jessica Houston, Professor of Chemical and Materials Engineering at New Mexico State University and President-elect of ISAC, and we discuss the difficulties of finding your feet as a new PI.
Sometimes finding your own niche as a faculty member can be tricky because you don't want to do what your advisor's doing, or you don't want to, be perceived as you're copying or piggybacking or whatever.
Right through to her successful Fulbright scholarship application.
I applied and got it. I didn't think I would get that grant, but I did get it on that first try. And we told our kids, look, we're moving to Japan for 6 months.
And we go back to the start of her career to hear about her early switch from industrial engineering to chemical engineering.
To be honest, that experience made me think, nope, this is not what I want to do. I don't want to walk around a plant with a hard hat, and this is not for me. So at that point, I looked at research opportunities in the department.
To how she was able to balance work and family life more recently.
We really have had important support over the years with my in-laws, as well as my parents, as well as their aunts and uncles that help. So we have family that can be with them if we can't. For example, both of us are traveling.
All on this episode of Flow Stars. Hi, I'm Peter O'Toole from the University of York, and today on Flow Stars, I'm joined by Jessica Houston from the New Mexico State University. Oh, and should I say, President-elect for ISAC. Jessica, how are you today?
I'm doing really well. Thanks for having me.
No, thank you for saying yes to coming on it. President-elect.
Yes.
That's a big deal, but the big deal's to come.
Yeah, it's a big role. It's going to be a lot of work, but I think I'm geared up for it. I've been with ISAC for a long time now, and I guess I could see it coming, but I didn't, you'd never expect it, but here it is.
Well, very many congratulations. It's a very honourable position as well, a very prestigious position within ISAC. For those who may be not familiar with ISAC, do you want to just describe what ISAC is?
Yeah, so ISAC stands for the International Society for the Advancement of Cytometry. Maybe a lot of people watching would know, but we are about a 2,000 person society. We are composed of a lot of members that have interest in quantitative cell sciences. So that ranges from people who are working in core facilities with flow cytometry instrumentation, and people that are in academia studying or utilizing flow cytometry as a tool to understand something in their, biological systems. People that might be more clinically inclined, that are utilizing it as a diagnostic means for things like immunophenotyping. So it's a society that welcomes all people that are interested in that area, in this big field. And we have a council, we run the annual Cyto meeting, we have a journal, Cytometry A, so we do a lot of stuff.
I just thought, you said there are lots of sections, lots of working groups. I bet you don't have an answer for this, so this is maybe unfair, but do you have a feel for how many different people are actually helping via the committees and sections?
Yeah, wow, that's a good question. I should know the number because we have a lot of volunteers. I mean, if you look at our committees, there might be about 30 or so we have, and within each committee, you may have about anywhere between 5 to 15 or more people participating, depending on if it's a larger task force or smaller. And so it's a lot of, and there's a lot of overlap. Some people are on many different committees, but I'd say we have basically a good body of volunteers. And I wouldn't know what to guess in terms of numbers or percentage relative to the overall membership. But it's certainly a lot.
Yeah, just working on those guesstimates, you must have 150, 200 different volunteers, which is shows the community spirit, I think, and the community willingness to support ISAC.
Yeah, right. It is. It's very community focused. And, the times that I've when I first started with the society to where it is now, I've noticed, that there's always a group of people that are always coming in, always interested. And once they get in, they seem hooked, hooked on it. Like, oh, this is a great society. I like the people in it. I like, the meeting. And so I want to be part of it. And so I want to join this committee or that. So yeah. And the fact that it being international, very much international, you would think that makes it a little more clunky in terms of how to get people together, but it does, it works. And, we're seeing now a lot of expansion to other parts of the world, a lot more expansion in Latin America, which is a great thing. So
I'm just thinking about all those committee meetings where you have people in Australia, Europe, America, and someone is always going to be rather inconvenient, but everyone still turns up.
People show up. In fact, we had a committee meeting yesterday, and we had somebody who was, on at 2:30 a.m. So that's dedication. And I mean, I lived in Japan for some time with my family. I did a Fulbright, and I remember thinking like, oh, great, I'm going to have to do these like ISAC committee meetings. And yeah, I would just get up early. I would be up at like, 4 a.m. or 5 a.m. But you do, and we have to work and maybe make a compromise sometimes, have meetings at different times to try to work with the time zone, but that's what you do.
I'm just thinking, we'll come back to ISAC in a minute, but actually before we leave ISAC, I'm just thinking back all the past presidents I can think of. You will be one of the youngest ever.
I might. Somebody brought that up to me, and I thought, yeah, probably. We have Johnny, and then Rachel, the most immediate, and then Paul Wallace, and we had John Nolan. I don't think, maybe he was closer in age to me when he was president, but maybe, perhaps. I don't mind saying my age. I'm 45, so so maybe, I don't know.
Yeah, I do. I was just going back with Paul Smith, Andreas. It goes
right. Andreas.
Andreas close to that. It's got to be very close to that. So then, so this is 45. There you are, becoming president of ISAC. But what got you interested in science to start with?
Yeah, so that goes back to high school. I think I was always, inclined in more of the STEM fields in terms of like interest area. So I was not very much into arts or, history or those types of social sciences. I was more liked math. I think I did well in math. And I really, in high school, had a great biology teacher. And so we have these AP courses, which you could get college credit for. So I tested into a bio AP class, and I just loved it. And she, at the time, she put me in touch with a local I grew up in Santa Fe, New Mexico. So in Santa Fe, there's a little bit of biotech, but Genzyme was one of the companies that was, that had a headquarters in Santa Fe, and they were doing samples, amniocentesis samples, and they were just basically doing genetic profiling with samples that they'd get from women all over the country. And so my teacher set me up with an internship there at Genzyme, and that's where I learned cell culture. As a high school student, you learn like cell culture, what it looks like to do, a little bit of genetics stuff. And so that was fun, and I was really into it. I ended up being engineering, not biology. So
I was going to ask that question. So at the age of 16, 17, where did you see your career going at that point?
Yeah, at that point, I just knew I was going to college. I just knew I was going to go to the university. My family, couldn't afford to really pay or help me get to a really out of state, more elite school. So I ended up coming to the state school, which is, NMSU, New Mexico State, which is where I work now. But I was an undergraduate here. And at the time, my dad, who was an engineer at Los Alamos National Lab, recommended to me I try chemical engineering because it kind of is somewhat close to bioengineering, which is what I started getting interested in and started learning about. Of course, I was learning what genetic engineering was, and then and this was in the mid 90s, and I was basically starting to kind of get a sense of that field. And I ended up joining the chemical engineering department and learned that it was completely different than bioengineering because, it's basically thermodynamics, it's fluid mechanics, it's really at that time was still focused on teaching students to be prepared for industries like oil and gas and chemical industry like Dow Chemical, DuPont, Chevron, Exxon. So it was really, it's like a curriculum made for students that are going to be process engineers. And so that was kind of eye-opening, but I stuck with it because I knew that I liked the challenge, and I just knew there was really nothing else I could think of that I would want to do. So I did it. Yeah.
So at that point, were you thinking your career could possibly go into the more chemical
I did.
area with DuPont?
I could have been working in a bleach factory, or I actually did an internship with Archer Daniels Midland, ADM. That's a huge company in the Midwest, and I lived in Illinois, and I worked at a corn plant. That's where they took corn and they isolated different parts of the corn to use it for different things. For example, they had these enzyme columns, they were using it to make high fructose corn syrup. So it was like a big, and they also took out protein to do something with the protein of the corn. So it was really a huge plant, and I did an intern there. But to be honest, that experience made me think like, nope, this is not what I want to do. I don't want to walk around a plant with a hard hat, and this is not for me. So at that point, I looked at research opportunities in the department and started in research.
I love the idea of working in a career in bleach, which
Right,
surely can never be a career. Your career must fade really fast if it's in bleach. So that's a really
I know, and because I had chemical engineering friends that did that, and they were telling me about their job, and I was like, no, I don't think so.
Yeah, very sterile. Anyway, so too many bleach. There really are too many bleach jokes to go through this. So what got you back into the light? So obviously that wasn't for you. So you didn't want to wear the hard hats. You obviously fancied the white coat and goggles instead, and blue overshoes. So how did you get back into life sciences?
Yeah, so in our department in chemical engineering, we had a faculty member. Her name was Sarah Harcum, and she was doing a lot of interesting stuff with E. coli as a model system. So she was using E. coli as a model system, and she was doing some things that were doing treatments of amino acids in the E. coli, and all of her whole project the focus was, if I can recall, a kind of a bioprocessing focus. So her objectives were, developing the model system of E. coli to enhance their ability to be used as in bioprocessing. So whatever that might be, because I think, chemical engineers at the time, they were in bio. Like bio was a big part of chemical engineering, but it was a big part for like scale up, for example, like using E. coli to make insulin, right? Or big fermentation processes where you use biological systems to make something, whatever engineering you're doing in the genetics to produce whatever protein outcome. So she did that research here in our department, and I began working for her, and I thought it was really cool because I was doing like these mini preps and just learning a little bit more about like, bio as a part of chemical engineering. And so after I worked for her, then I knew I'm going to go to graduate school. So at this point, I know that it's going to be a graduate school trajectory for me, not just an industry job.
Yeah. So where did you go for your PhD?
So then I went to Texas A&M. So Texas A&M University in College Station, Texas. And actually, there's a reason I went to Texas, and that's because at the time I was getting married, and my husband, who was also he was in biochemistry, was already at UT getting a graduate degree. So he and I met at NMSU, and then he moved to Texas. We lived apart for some time, and then I knew I was going to graduate school, so I looked around at universities and found Texas A&M, which a program I really liked, and I ended up doing some really cool graduate work that was what I had not expected at all, like a total different field, and it was all in a chemical engineering department, but that's when I started branching into optics and more optical engineering stuff.
Okay. So actually, I think I don't know. I presume this is your husband on this picture?
Yes. So that's Kevin.
For those who are listening, can you describe the picture for us?
Yeah, this is a recent picture we took only a couple of weeks ago. We went to Houston, Texas to watch the Houston Astros play, and that's the game. That's Kevin, kind of in the between two of our sons. Joaquin is on his right and Caleb's on his left near me. And then our daughter, her name is Kira Luna. She's there too. But we have this big love for Houston because, I can tell the rest of my story, but we ended up living in Houston for some time before we moved back to New Mexico. But we're big baseball fans.
And that was Baylor College of Medicine that you went to?
I was there. Yeah, I was at Baylor. I did some time also at MD Anderson. Yeah. Yeah.
So okay, so you were getting into optics, interested. We're still not at flow cytometry yet.
It wasn't flow yet. It was not. And so I was in graduate school in the very early 2000s. And I found this advisor in the chemical engineering department. Her name was Eva Sevick. And Eva was one of the greatest advisors, I've ever worked with. She was building this research program, all focused on the area of frequency domain photon migration. And what that basically is using near-infrared light as a means to interrogate dense and deep tissues. Mm-hmm. And so as we know, near-infrared light has a propensity to go deeper in the tissue because there's more scattering and less absorption in blood and melanin and water, so it can scatter deeply. So back then she was developing this, and we call it frequency domain because we were modulating the laser at radio frequencies and measuring the propagation of near-infrared light into deep tissue and then detecting the light as it propagated back out as a means to try to characterize the tissues itself. So let's say it's a tumor, the heterogeneity within the tissue. And it's a challenging field because, as you know, all of the imaging modalities out there, whether that be X-ray CT, MRI, ultrasound, do a much better job because they can actually, those sources of radiation will very nicely be detected deep, and whereas light is difficult. So, but we were developing a tool to do that. And she started working with people at MD Anderson so we could do this in small animals. And then she eventually went to Baylor where we were doing actually clinical studies with women to try to detect. So that was where I started, learning a little bit more about biomedical optics, about fluorescence, contrast agents, drug delivery into deep tissues, using light, using light and lasers and detectors and all of that. Yeah.
Still not on a flow cytometer though. So where was your first interaction with the flow cytometer?
So that was as a postdoc. So then that takes me to after we graduated, Kevin and I decided to start a family, and we wanted to come back to New Mexico because we're closer to our families, and we wanted some extra support. And being that we were both had a PhD, there, the state of New Mexico doesn't have a whole lot of industry for PhD. We have our national labs, we have our universities, we have some biotech kind of happening, just not a lot, but we knew that Los Alamos National Lab would be a place where we could both work, and we found positions there. And that's when I first got into flow. Interestingly, when I interviewed at Los Alamos, I was interviewing for a job in the weapons program. So there's two facets of Los Alamos National Lab. There's the side that does more science and engineering, and then there's the side that's very much focused on the nuclear non-proliferation stuff and what we would say weapons. So I was interviewing for a position in a division there, and a person from the National Flow Cytometry Research Resource, the NFCR, at the time, came to my talk, and he said his name is John Martin. And I'll never forget, John Martin went back to the director of the Flow Resource at the time, was Jim Fryer, saying, Jim, you might be interested in this person because she's interviewing for this job, but she would be fit really nice in the NFCR. And so that's when they reached out to me, and I said, oh, I didn't realize there was this big flow cytometry group and, people that are doing biomedical optics, which is right up my alley. And so that's when I switched over and got the job there. That's when I started flow. So that was probably 2006. And at the time, Jim Jett was still involved in the flow resource, but it was Jim Fryer who was the director.
Okay. So is this a picture from back then? I haven't got a description on this picture, but
That's a picture from graduate school. So that's when I first, was doing stuff like that with, okay, we got an oscilloscope, we got a function generator, modulating laser, that type of thing. I never thought was chemical engineering, but I was in this chemical engineering lab doing that stuff. And you would think that it would be more physics or electrical engineering, but yeah, that's graduate school. And I think there's another one that I have it from Los Alamos.
I'm going to have to guess which one that is now.
Yeah, there we go.
Ooh.
That's the Los Alamos pic. That's the lab that I worked in as a postdoc. So right behind you is a big flow cytometer. Can you see the big laser? There's a, right by the door behind you, there's a long spectrophysics laser, an argon ion, yep. And there's a flow cell there. There's there's a function generator, some oscilloscopes. There's a lot of mixing boards there that we were doing some analog mixing. So I worked on this tool that was a what we called we called it the phase system, but it basically is a lifetime flow cytometer. The flow cytometer that can measure fluorescence lifetimes. Yeah.
Wow.
I noticed the cables going over to the right or over. Those are cables that connect into a data acquisition system that was built at Los Alamos. So we had we were trying to do real-time analysis of lifetime, and that instrument was actually, when I got there, it was kind of starting to collect some dust, and I said, what's going on with this? And they told me the history about it, and the history is really great. I mean, that instrument was originally designed and built by John Steinkamp. So Dr. John Steinkamp worked on it. He's really the brainchild of lifetime cytometry, and Harry Chrisman as well. So him and Harry Chrisman, and I've met both of them actually, and they came to Los Alamos during the time I was there. And they both were used it a lot. Harry had a lot of interesting applications for measuring lifetimes with cells. And John was really, a lot of the engineering and developing and he was so meticulous. His lab notebooks were in that lab, and I would read through his lab notebooks, and they had these like perfect descriptions of what he was doing, and like very, very careful calibration and optimization of that instrument. It was really interesting. Yeah.
Okay, so for those listening, the picture isn't anything like you resemble a flow cytometer today. There are more, it looks more like a telephone circuit switchboard with more cables coming in and out. And a ton of spaghetti, black spaghetti of the wires, absolutely everywhere.
Yeah.
By the time I got in, I was in the MoFlow generation where there was a switchboard and wires, but nothing as complicated as that looks.
Yeah.
But I suppose quite literally that was a lifetime ago. Oh sorry.
Good dad
Just
joke,
one of those
right?
days. So what was your first commercial flow cytometer? Can you remember?
Yeah, it was a fax caliber, because we were right in that same lab. You don't see it, but on the other side, Los Alamos by that time had acquired a lot more modern. We had an LSR2, we had 2 calibers. They had an old Vantage. What else were they getting at the time? I think they ended up getting an Accuray. And so there weren't a lot of really the new stuff until I left, and then they acquired more new stuff, because by now I know they have kind of a Sony system. They had an imaging cytometry system. But we were still at the time really working using the caliber a lot. A lot of the users in the division were running samples on that. I'm trying to think if I'm missing one of them.
I don't know. Caliber was my first, and then I went to Cyans, actually. So the first Cyans that came out, which were revolutionary at the time, really small and super, super easy to use. Certainly in our hands, they were very reliable in that case. So you're now not just involved in the academic side of the optofluidics, the bioprobes, fluid dynamics, which all those skills you've brought up, you're brought up with through your PhD postdoc research days, are now still with it. But you also run a core facility there as well.
I actually don't really, because at our university, we don't have a lot of people that necessarily use it. I mean, we're a state university. We're engineering ag strengths. We're kind of the land grant university, which means we serve, all the students from the state. Our university is about roughly 14,000 students. It's not huge, but in general, we don't have we're not near we're near an osteopathic medical school, but we don't have a medical school on campus nor a vet school. So there isn't the critical mass of users to really create a cytometry core. We do have a microscopy core on campus, and then there's kind of another quantitative science core where we have lots of mass spec and some GC stuff. But what I do with my cytometers, because I've acquired them over the years for my own needs, is that I just welcome users if they need. So for example, there's a biology professor and he reached out to me recently. He's like, can we use your flow systems? And we'll train them on them. We'll help them with it and then they can use them. So that's kind of how I've done it. And usually it's not overwhelming. There's not a lot of people it wouldn't be manageable, or we would need kind of the more infrastructure that a resource lab would need, like the key training people and the educational component and all that.
That's very similar, I guess, of how core facilities would have started, because there wasn't a user base necessarily for them. They would have had it for their own primary research or development, R&D, and then the academics start to come and want to use it, and there's that bills. I just watched this space. Yeah. They must have more biological research there, just needing the bacteriologists, the yeast, the fungi, the algae, beside the cell biologist. Surely they're using the flow cytometer. So yes, you need to get them back in there and using it.
Right. In fact, I mean, even the people that I work with and, I'll collaborate with people or just in other realms of the university, and they're like, oh yeah, I know, I want to use your flow for this project or that. And then, people get so on a track of what they know. And then, you got to sometimes breaking them away or really letting them recognize, hey, look, this could be a way to characterize what you're trying to do.
So you sent another picture, which is probably apt at this point, of yourself.
So that was taken with my very first PhD student. So I was a very junior faculty. I was starting off, and I thought, what am I going to do, right? You're in this faculty position now, and you have to run a research program or have, this bright, brilliant research idea to write grants to get money for it. What I did is I told Jim Fryer at Los Alamos, I said, look, I want to continue lifetime, and the only way I can continue lifetime is just to basically replicate what I was doing there as a postdoc. And he said, go for it. Sometimes finding your own niche as a faculty member can be tricky because you don't want to do what your advisor's doing, or you don't want to, be perceived as you're copying or piggybacking or whatever. But he wasn't, nobody was really doing that at Los Alamos except for me as a postdoc. And at the time in Los Alamos, a lot of our members were kind of going to branch off into other things. And so I took some of the instrumentation down with me to NMSU. They gifted me, and I started building a lifetime cytometer there. And we built it with my graduate student, Rofan. He's now works at a microscopy core in Mississippi. But he and I started assembling that. I had a lab full of nothing. I came to NMSU and they said, here's your lab, an old sink, nothing there. So I had to take this old instrument and begin to rebuild it and basically develop a cytometry system.
So he was your first PhD student. So a small group starting back. How intimidating was it to start your own academic group and have your first students and so forth? How did that feel? That's quite a big step.
It's very scary. It was, you feel like a deer in the headlights. Like, what do I do now? You just know the expectations, right? And the expectations are to bring in lots of grant money, to publish as much as you can in high impact papers, to get your students graduated, past a master's, past a PhD degree, to teach all these courses that we need you to teach, to participate in service activities. So you have all these responsibilities, and all you can do is just time manage as best you can. And so that's all I did, is I just tried to do that, and then there's the lingering fear of, what if this project doesn't work, or what are we going to do next? Or how do I know if I'm going to come up with a good idea, bright idea to take the next step? And that's when you rely on your student and how they are and their, their ideas, and you guide them through that, and you help them read the literature and papers. And it's tough. It's tough. You wear a lot of hats. The mentorship hat is really tough, because getting students to learn what it's like to be a researcher, to do a PhD degree. But I've always surrounded myself with people that are really helpful. Like, I continue to collaborate with some people from Los Alamos. For example, Mark Niver, who is now at Miltenyi Biotech. He does all the data work analysis stuff. And he's been great. He's like been, big part of like the stuff we've done, because it's the data system that we really rely on for lifetime analysis. And just other people in general, like even Kevin, my husband, he has a lot of good biology that we could use as applications for the lifetime instrument. But yeah, it is, back to your question, it's kind of nerve-wracking and stressful.
You had the N of 1 at that point. Your group has grown. So I presume this is your...
So that's kind of a more recent group picture. It's kind of gotten bigger and smaller over the years. I have a postdoc there on the end. Jesse is in the black shirt. And then I have undergraduates. I have 2 graduate students in the front, Samantha and Arik. Arik just graduated with a master's degree, and he's working for Cytech now.
Wow.
So he's the guy right in the middle. And then the people behind him are some undergrads. And then I'm kind of hiding back there too. But yeah, that was a more recent picture of my group. In the past, I've had bigger groups where I've had like 4 to 5 graduate students, a postdoc. I've had like sometimes as much as 7 undergraduates. So, and then now, right now, my group's actually kind of small. I only have 1 graduate student, 1 postdoc right now. But there's always undergraduates that want to do research. So they can be really helpful, because they're all very smart, but it takes a lot of work to train an undergrad, because then they do research with you for a semester, and then they say, okay, thanks, and then they go on their way. So the investment in time is tough with undergraduate students.
Yeah, no, I fully appreciate that. The time, effort in to get the reward back, which doesn't always follow. But yeah. And so thinking of undergraduates is also the importance of teaching, and not just teaching undergraduates necessarily, but teaching more globally. So yes. You sent loads of brilliant pictures. So could you describe what's in this picture for those listening?
Yeah, so one of the university was doing kind of a promo on active learning. And so this is one of our classrooms on campus. That's what we call an active learning classroom, because the walls are whiteboard walls, and the students sit around in round tables, and they each have like a monitor and laptops for each of their round tables. So it's geared more for them to be doing something rather than just sitting passively listening to a lecture. So what I was doing there is I was having them do teamwork, and then I was walking around the classroom, helping them as they were doing on the whiteboard, solving what we call the Navier-Stokes Equation. So in fluid mechanics, there's a big momentum balance that you do to understand how velocity profiles change and pressure differentials change. So like, for example, like how airplane gets its lift, or how we use it to characterize also flow through a pipe. So water flow through a pipe. So that's the basis of laminar flow, right? And laminar. So I was working on an equation there where we were learning how to balance momentum, or it might have been an energy balance on that one. But yeah, I do a lot of teaching. That's part of chemical engineering, is teaching different classes.
I'm thinking now, you've got a lot of teaching. Your group fluctuates in size, but it can be 6, 7, 8 people in your research group. You're writing grants, you're publishing. You've said yourself, you're only 45, and you have 3 children.
Yes.
And the ages of those, if I may ask?
Right now, they're 15, 13, and 10.
So a lot of this career has been developed alongside balancing a family life as well.
Right.
How have you gone about that? Any tips and tricks for people there thinking, well, I want a successful scientific career like yourself. I want a family. How do you balance all that?
I get that question a lot, and it's difficult to answer because I don't know how. I think I take it day to day. It doesn't come without sacrifice. There are times I've traveled. In fact, the last meeting in Philadelphia, my kids were like, "Mom, why can't we go with you?" And Kevin and I just said, "Well, it's just a quick trip. I'm going to be busy the whole time." And so there are times I'll have to leave them, but I'm thankful for the family and support, because we really have had important support over the years with my in-laws, as well as my parents, as well as their aunts and uncles that help. So we have family that can be with them if we can't. For example, both of us are traveling, and over the time of, going through tenure was tough, but we, if we're not at work, we're just home with them. It's, you have to just know that there's not a lot of like your own personal time. It's just you're with family, you're with your kids, or you're at work. And COVID changed things a little bit because we were home a lot with them, and we stayed home with them, and they liked it and I like it too. And I technically could do a lot of stuff from home. I can't be in the lab and like watching the students, but I can do stuff from home with the kids there. And now they're older, so they're more self-sufficient, and they don't really need the hand holding. But I have to owe it a lot to Kevin, my husband, because he was really stepped up. He's not quite as busy as me. I know his career's kind of been a little bit different. He does he's a biochemistry professor. He does a lot of publishing and grant writing and all of that, and he's like an associate department head, but he's not much into the society work like I am, so he doesn't have that stuff that he's doing.
That's a good question, actually. How much time would you say you actually put towards the society activities? Do you have a feel for that?
I'd say it's at least 25% of my time. It's just a quarter of my time, because if I look at my day, I say at least, it could be even more during some parts of the year. When I'm teaching in the fall, that's usually like 50%. And then like reading, doing research should be at least 60%. But by then I'm over 100 here. But I think with the society, I put in at least, like, for example, yesterday, I think I put in like 3 to 4 hours with ISAC, doing meetings, doing some planning, doing emails. Yeah.
So that's on top of your 100%.
Yeah, it's on top of what I should. Well, some things do suffer. For example, I've been wanting to write 2 grants and I just haven't. And I just feel like those are the things that can easily be put to the back burner, but then it becomes worrisome because there's no deadline for those. And so you just think, oh, I'll do that, I'll do that this summer. And then all of a sudden it's July. Okay, well, maybe I'll try to do that for the fall. And that's worrisome. And even papers, like I rely a lot on my students to send me a paper so we can talk about it, work on and submit it. And this year I've had a paper deficit, and that's worrisome too, because I know that and a lot of that's owing to kind of COVID, coming back from COVID, but also a dip in like the students I have right now and who's working, which is fine, but still that's worrisome, because if I'm not publishing one given year, then I have this gap on my CV where I don't have papers, and that's not good. And so
I think over the COVID years, I think that the on the grant panels, you're going to be looking at CVs and thinking, well, obviously there's a gap. It's going to slow. It's going to thin out at the very least.
Yeah.
Over that point,
true.
And that's just, I think, the way it goes. So, as you say, it's a lot of hours. And you said you like baseball.
Yeah.
And traveling.
We like traveling. We like baseball. Like that's a picture in Japan. So a few years back, I had kind of a grant rut in which I wasn't funded, and it's before I got my R01 grant, which my NIH grant now, and then I had some NSF money and they expired, and then I said, I'm really like wanting to do something different. And so I learned about the US Fulbright program, the Fulbright Scholar Program, where faculty can apply to the State Department for a Fulbright grant and then be paid to basically live in another country for some time doing research. And at the time, I started collaborating with Miho Suzuki from Saitama University, just north of Tokyo. And I reached out to her and I said, I'm going to apply for a Fulbright to see if I can work with you in your lab, because she had some really interesting stuff that she had an application for lifetime cytometry using like FRET. So she had this FRET probe that would go into the cell, and she would do some enzyme cleavage and lifetime is a good tool for that. So I applied and got it. I didn't think I would get that grant, but I did get it on that first try. And we told our kids, like, look, we're moving to Japan for 6 months. So that's another nice thing about a faculty position, is you get these opportunities, it's a flexible, and you can leave your university for some time, and you can still be paid, but you now you have this experience.
And any concerns about taking the children out sort of 6 months, a long time out of their, they're a young age, they're in education. Was there any concerns there? Were they worried about it, or?
They were worried, but they trusted us, and we were certainly worried. And fortunately, it was a good time of their schooling for us to do that, because nobody was in the middle of high school yet. They were all still kind of younger, kindergarten, elementary age, and so they weren't in middle school yet. So they were all right. They could all be in the same school. But it was an experience. They loved, at the time, they didn't like it, but now that we are back, they talk about fondly, like, we want to go back to Japan. But it was hard for them at the time because we put them in the Japanese public school system.
Yeah.
And they just didn't, they didn't know any Japanese. We had no language skill at all in Japanese. And they just were immersed in that, listening to a teacher teach them in Japanese all day long. And they started picking it up. They would get like some assistance with some teachers that were, English, but it was tough for them. But we made them do that, and they agreed, and they were worried, but they trusted us, and we said, hey, this is a good experience for you guys. So they would walk with all the Japanese students to the school every morning. And it was neat to see.
It sounds terrifying to have that sort of just dropped in. And so how is their Japanese?
They were literally dropped in. So right now they have no knowledge Japanese. Our youngest, she does very well counting, because in kindergarten they do a lot of counting and like kind of using like the hiragana, katakana kind of Japanese phrases. So she does okay, but none of us really picked it up. During the time they did okay. They could understand kind of what the teacher was trying to tell them. And there were things that we remember, like how to say, things like, well, hello, how are you? Or I'm from US, or things like that. But it's hard. Language, unless you're there for years and years and you keep up the communication, as they say, you'll just lose it.
So I'm going to come back to some other things now. So I've asked how you got into flow cytometry, where you are today. If you could do any job in the world, what would it be?
Wow. I think I would be a stay-at-home mom. I know I consider that, it might not be like a career, right? But I think that if I could just go back and have the choice or ability to do what we want to do, like all of this traveling, all of these experiences with them, but be able to just stay home with them, I would have done that, I think. Because you do miss a lot of their life when you're working a lot. And it's hard to imagine, like a different career outside of what I'm doing now. I don't know, but I think, being just like the at-home person would be nice.
But in 10 years' time, they'd have fledged the nest, and they won't be at home anymore. So
true.
And you have a really good career.
Mm-hmm.
Would you call it a job? Would you call it a career, or would you call it your passion?
I would call it a career. It's somewhat become a passion. I mean, somewhat, because for some reason I found this area, this field. I found like a community of people that I like to work with. A thing that I'm kind of good at, we stand out, we have our own little niche within that field, as kind of an expert in that. And then the leadership stuff, participating with people to run the society. So I think it's a passion from that perspective, in that I thoroughly enjoy it. I really do. I don't have any complaints about it. But I wouldn't say like it's like it's like I couldn't live without it. It's not quite that, but certainly a great job. I mean, being an academic, teaching, you have new students every semester, fresh, blood that or fresh students that are like, really eager to learn. They keep you kind of young at heart. The faculty you work with are all kind of focused on the same mission of improving the curriculum and teaching. Then aside of that, I have this entirely other people that I work, other group of people in cytometry. And so it's a great, I don't know, it's hard to see myself doing something else, but.
I'm glad you said passion, because I can't see how you would not be doing so much for ISAC if it was not a passion. I think it has to be, because so much is voluntary.
Yeah.
You're choosing to be there, you're choosing to drive it, which actually comes to a good bit. Mrs. President-elect, what's your agenda? What are you going to do? Do you have a vision? Is there something you'd like to pick up on and drive through in your? It's quite a short term as well, isn't it?
It is, and so the role of president-elect is actually to to run the meeting. So that's kind of the most immediate thing that I'm going to be working on now is chairing the entire program for Cyto Montreal. Yeah. So I'll be focusing on that, and that's a big job, right? And we, need to make sure that all of the people that want to participate can participate. Everybody has a voice and what they think the meeting should look like, and what they think, the speakers should be about, or the plenary sessions or the parallel sessions. So that's a huge, thing. So that's my most immediate, task ahead of me right now as president-elect. But also president-elect eventually starts working with all of the committees and chairing things like one of our committee collectives, which, getting all the committee chairs together. There's a lot of, you're part of the executive committee. So the executive board is basically, past president, president-elect, president, and then, the other officers. So there's that. And then once I am president, which will be a 1-year term, and that begins in 2024. So that'll be a couple years out.
And I've got to say, so far, we talk about lots of volunteers, but it's not just about volunteers. The staff that are paid. The conference costs a lot of money. There's a lot of online content which costs a lot of money to produce, publish, put online. It's a big responsibility, and the society is big. And COVID must have posed big challenges, because as you say, you're responsible as chair. So I've been here with the Royal Microscopical Society and the big meetings. If the meeting at Cyto is a huge liability, it's probably not the right term to use, but if it was to go wrong, it could almost fold ISAC if Cyto was to go wrong. What things are being? How have you coped over COVID? Because you were treasurer as well.
Yes.
So you'll be very sensitive to these finances.
Right. And, some people would tell me, like, how do you sleep at night? And I never thought we were like in this dire strait where we were it was going to break us. I mean, we knew that COVID, is affecting people worldwide. We knew the economy was going to be going crazy for us, because that's where investments are. Basically, we knew without any meeting, we were going to take a big hit. Luckily, over the years, we've had some, nice buffer for that. But we also were going through a time at ISAC where we were transitioning away from a managed society through FASEB to self-managing ourselves, which is basically hiring our own staff and all of that. So there was a lot of cause to worry and to be fearful of what is going to happen. Are we going to survive this? What will ISAC's finances look like in the future? And so the things that we did were tried to be as conservative as possible. We knew that we had already created a budget. Our annual budgets in for things like the meeting, budgets in staff time and effort, and all of that. So when COVID hit, things happened in which, okay, maybe we're not going to get all this meeting revenue, but we already have in our budget, and we know what the cost is going to be. Maybe we can get away with a virtual meeting that's somewhat lower cost, look to our industry and vendors to support it, and then carry through. And then there's a philosophy of how much should be in your reserves, how much investment should you have to save the society if something is very critical. So at that point, Johnny, our president, came up with a mission continuity committee to be thinking of and focused on situations that could occur, and we need to figure out ways to mitigate those. So it was a tough time, yeah, but we would have lots of Zoom meetings throughout COVID with Andrea Kusturica, who was the president, and then Johnny, who was becoming president. We had Paul Wallace, past president, and then myself and as the treasurer. So it was, and then David Galbraith was secretary. So we had a lot of meetings to think about what's going to happen, and.
It's interesting. I guess because you were under FASEP at the time, for the Royal Microscopical Society, it's had that. It's got its reserves to weather storms, but it's painful, when you're sitting on its deck to actually see those funds dropping, because that's your rainy day money.
Yes.
On a rainy day, and it's going down. And you think you can't only have so many more rainy days. Fortunately, I've got to say, I think similar for ISAC. It was very much the damage was very limited, and I think again one of the great confidences, and I think you say you're not losing sleep over it, because the membership is very supportive, so they will get behind whatever is being delivered, and they'll be registering for the virtual meetings, whatever else it is. The companies are very much on side as well, because they're very much part of the same community. And I think it's really important to have them completely on side and as transparent as possible with it.
Right.
It'll be interesting to see how it goes, especially because I know the membership model, so the fees model has changed, and it'll be interesting to see how that pans out, because that looks quite experimental from where I'm sitting. It'll be interesting to see how many of each tier come in to see if that budget actually does work or not.
Yeah, when we were thinking and rethinking the membership tier structure, a lot of research had to go into what are other societies doing? How is this going to work? What do we anticipate? What structure do we anticipate most of the people opting into?
Yeah.
How can this support people from countries that don't have the infrastructure, or that may have a more income that's not, that wouldn't really allow them to come in at a platinum. So how do we use that as a consideration? Because this is a worldwide society, and this tier structure is going to affect everyone. Interestingly, I think what Courtney, our ED, said is that we have more people doing the platinum and gold than she expected. I think the highest participation rate is in the highest tier, and maybe one of the third down tier. She thought a lot, we were expecting a lot more coming in at the lowest tier, which is a cheaper, way. But the way that people are actually be registering as members was a little bit more unexpected. So
it would be interesting to see how that breaks down globally, because I think certainly in the UK, the platinum model will always be almost impossible to get through, certainly work expenses, to say, because it's inclusive of the conference. It would be hard to actually justify that from a UK and maybe even European perspective. So it'd be really interesting actually to see how that model works in different parts of the world itself. I need to move on because we've only got 10 minutes left. I'm just very quickly through. Some quick fire questions. Are you an early bird or a night owl?
I would say I'm a night owl.
Okay.
Yeah, I can work at night better. Yeah.
PC or Mac?
That's a funny one because I do both. I have a Mac. This is what I'm on right now, a big iMac, but my laptop's a PC because I need certain programs in chemical engineering that use PC. So I do both, and I love both.
Okay, so let's answer. Okay, so McDonald's or Burger King?
McDonald's.
Oh, that was a quick answer. So what's your go-to? So, okay, there's not many ever say that. So what is your go-to when you go to McDonald's?
The sausage egg biscuit.
I don't have that in the UK. Okay.
It's a breakfast sandwich. Yeah.
Oh, yeah. Actually, breakfasts are my favorites. I've got to say, I think McDonald's do a pretty good breakfast. It's different. It has its place in the
And their coffee. I really like their coffee, actually.
Yeah, no, their coffee is good. I wish they did decaf. On that note, tea or coffee?
Coffee, yeah.
Wine or beer?
Wow, that one also is very tough because I like we have a microbrew program in chemical engineering here in our department, and there's a lot of microbreweries all throughout New Mexico.
Isn't that the protein
very
fermenters?
popular. Huh?
Are you sure that's not just a protein fermentation lab that's doing?
Right? See, it's a chemical engineering problem, like, right? But there's also a lot of wineries around here, and I do enjoy wine, so it's tough. Maybe probably beer if I had to choose.
Okay, chocolate or cheese?
Wow, cheese, I'd say.
Definitely. Okay, go to a conference. And maybe you're an invited speaker, and you get taken out for an evening meal. What would be the ideal food that would be served up for you? Because quite often you don't get to choose what you're going to eat. It gets put in front of you. What would be, if it came out and you go, oh yes, perfect. What's that food?
Probably eating like a Japanese dish. Like sushi's great, could be sushi, but it could be something like okonomiyaki. I love that when we had that in Japan, or takoyaki, or even yakisoba, like something like that. I would be like, this is great.
Okay. On the opposite to that,
yes.
What is the worst thing that they could put in front of you and you go, oh no, and I have to eat that?
Yikes. Oh gosh.
Is there anything you don't like that you'd avoid
I'm
eating?
not a picky person, so I can't think. Probably something like if something bland, like just something like, I don't know, meat and potatoes. Like that's not to me interesting. Like I love a steak, right? I don't know, like, but I don't know, just I like things that are really more like ethnic, spicy, different. So something bland, I would say, oh, this is boring.
Who cooks at home?
We all do. One of my sons loves to cook. He watches this YouTuber and then my husband and I like to cook together. So we kind of all do it. Sometimes one or the other of us will like have more time to do it, but for the most part, it's a group thing.
Okay. And book or TV?
TV.
And so what's your TV vice that you're now going to confess to, but you'll regret afterwards?
Yeah, right. We watch so we subscribe to everything. We have Hulu, we have Peacock, we have Netflix, we have HBO, we have, what's the other one that I'm forgetting? So we always have a recurring show we're watching, and it's just a way to like relax. I feel like, I know some people can relax with a book, but I do so much other reading, like reading grants, reading papers, that when I go home, I just want to like stare at something. And so right now we're watching a Hulu show called Dope Sick. So we were like kind of binging that right now recently, but we were watching the morning show. Oh, Apple TV, that's the other one. We were watching that. So I don't know, we'll pick whatever is like the good show and we binge it.
Okay, that's a very good answer. And what about your favorite film?
My favorite film? Wow, that's a tough one. I mean, there's every year it's good movies that come out. I mean, like if I had to choose, probably like The Shawshank Redemption. That's a great movie, great story. But I honestly, I do love movies, and it can vary.
Okay, do you have a Christmas film?
Oh, a Christmas film. We love Christmas Vacation, Lampoon's
National
Christmas
Lampoon.
Vacation. Yes. We watch that every year, and like I'll always laugh at that movie no matter what.
Yeah, which is sad because you know what's going to happen, but it is still entertaining.
Right. Exactly.
I won't ask what your favorite scene is. I'll do that afterwards. What about you? What type of music are you into?
I like so I listen to like the the satellite radio stations like XMU, so like alternative stuff. I do like to go back to like alternative things like oh gosh, like I'll probably date myself, but we'll listen to like a lot of The Cure or New Order, or we'll listen to like Pearl Jam, like stuff like that. But I also like more modern stuff. Like I really like the Bleachers, and I like the Churches. I like Arcade Fire. So I don't know, we're kind of more, I feel like more like rock genre, I guess, alternative genre.
No, it's all good. There's nothing to be ashamed of in that. So what about when has been probably the best time in your career? If you could relive one year in your career, what year would it be? What time?
I think I would probably relive the early years becoming a faculty, because that was really fun. I mean, it's stressful and it's kind of intimidating, scary, but those early years, I just started as a faculty member. I hit the ground running. I wrote grants and I was getting these grants, and I got an NSF career, which is a big award for a young faculty. And I always just felt so great at that time in my career, like, okay, I can do this, like that's like the confidence you need, the confidence builder to say, like, this is a good career choice. So probably the early years as a faculty.
Okay. And on the code, which actually is really good to hear, because it is challenging, but it can be you've got a lot of freedom as well, which is very nice, and own responsibilities. What about the most challenging or difficult time you've had so far in your career? I'm not saying you're going to have more difficult or challenging times in the future, but in the past, have you found the most difficult or challenging time?
Yeah, I think one of the most difficult was like going up for tenure and promotion, because you start to really like be introspective of what you've accomplished, and you look and you can compare yourself to other people, and like what I've accomplished it does pale in comparison to a lot of people who are at universities like I don't know, USC, or I don't know, pick any big name, Michigan or whatever, MIT, Caltech, places that are very, very elite schools. And what I do is really, my productivity is not really anything compared to that. I have to say it's, I have a good career and I've produced work and I've published and all of that, but there are certain, levels of academics that, and I'm okay with that. But when you start thinking about that, and it's very stressful and it's not great, because then you have to put yourself out there to say, I'm applying for promotion. Am I gonna, make the cut? And then people have to evaluate you. So it's not a good time. It's kind of stressful. And
yeah, I will try and counter a little in your defense on this. If you go to MIT or Stanford, or they have big teams. They have good funding streams. The funding, they have a lot of PhDs and postdocs within their labs, so they can turn out more. I think what they do is amazing, because to manage that size team with such diverse research projects in the same focus field but so diverse, it's quite incredible. But I don't think it lessens what other universities are doing and the academics within their niche fields. It's equally valid and equally important. Yes, you're not going to be publishing the same volume that they have the ability to because of the number of students. But again, the influence is still there on the field. It's you're still teaching and inspiring. So no, don't worry about that.
And you're right, because we don't get the best students. We'll get like kind of the second tier international students. So, those schools draw the really strong, graduate student applicants that come in. And you're right, there's a lot more money, there's a lot more resources. And so it's true, it's probably easier to have those outcomes when you're in a situation like that. And
and if you're saying, if you're classing it as second tier, it's those who are coming in maybe with less opportunities to get into that point. You may have more influence over their future career than those at a higher university, because their career is maybe already trajectory into a degree. This is stereotyping, so we have to be careful. But generally, I would say it doesn't matter where you're teaching. You can still make a big difference to the lives of the students coming in. So no, it's all just different, metrics. And we are just gone over the hour. So I have to ask quickly, SPIE, SPI, or Cyto, which is your favorite conference?
Cyto.
You had to say that, didn't you? There was no choice. But SPI is also another great conference that you must go to on an annual basis.
Yeah, in fact, I chair with Attila. We chair one of the major sessions, the cell and forgot our name of our session. It's like the tissue cell session. So it's big, and I do a lot of work with them too. But yeah, I go to that every year, but
yeah,
it's a good one.
Jeez, I have to get over. Anyway, Jessica, thank you so much for joining me today. Everyone who's watched or listened, please don't forget to subscribe. Please don't forget, actually, a really unscientific thing, like it. We don't often do that. And someone pointed that out to me the other day, that everyone's very quick to criticize, not the podcast, but just criticize other things. So actually, yeah, come on, get out there and tell people about it. I think, Jessica, to hear about your career, how you've balanced it, the challenging times, I think it's been really good for people to hear, I hope, and show that it can be done and you can succeed, which is cool.
Well, thanks a lot, Pete. This has been fun.
Oh, and I'll see you soon.
Yes, see you soon.